opening up uses of our scanner quite literally, as you can see in this image here. So this is the system before our upgrade last year of the PET-CT, and usually there are people that are putting their heads in there. And we did this imaging over a couple of weekends in which we made very, very certain plastic wrap the scanner essentially to make sure that this type of radioactivity didn't contaminate everything else. But essentially what we're looking here is tracking the sodium dynamics. And of course, soil salinity is such an issue for Australia and the world in general, that it seemed like a really useful thing to do, to be able to take radioactive sodium and track it as a PET scan in the plants. And so there were different genetic variants here of the plants, and what we're interested in is how the dynamics of the sodium changed in the plants, and that led to various. I'm just going to flick up here to a couple of graphs, and you can see roots, experiment 1, experiment 2, roots, leaves. And the interesting things is for us were about how the plants actually changed their behavior before the lights turned on for the daytime. Okay, so I won't draw you to any particular one of these graphs, but essentially there is activity that shows that the plants have circadian rhythms that are independent or not completely dependent on when the lights are turned on and turned off, which was fascinating to see across a long time scale of a PET scan. And I'll just quickly show you the movie that came from these, from one of these experiments. So here we've got those 4 plants that are sitting in the PET scanner, and this is across hours, right? So this was about 70 hours of scanning. As time goes on, you can see that the radioactive sodium is being taken up into the stems of the plants and eventually all the way up into the leaves. And so we were tracking this over day and night to see how the sodium came and went at various times, which is useful to know if, and you should, you already might be looking at this saying, well, what was wrong with this one in the middle? Well, that's got a particular inhibitor, and this one here had, extra growth factors added to it. I'm trying to use my non-plant words to describe something that's actually quite technical. But you can see that this gives plant scientists a completely different way to do their experiments, because without access to the 3-dimensional imaging techniques, the way that they do it is that they have a whole bunch of plants and they add radioactivity to it. And then at different points in time, they go away and slice and dice the plants and do a radioactive detection on it, and then make up the same kind of graphs that we can do without butchering any of the plants over the time. I shouldn't probably have used the word butchering, but you get the idea that this kind of 3-dimensional imaging is really, really powerful way of tracking differences without having to set up such quite a tedious study of slicing and dicing and measuring in a very different way.